Tom's no stranger to the Department of Neurosurgery and the Brain Tumor Center, but just for those who don't know him, he's just an outstanding researcher. He runs a thriving research program in which the goal is to determine genetic mutations that underlie pathobiological disorders, neurological disorders in a number of different ways, particularly interested in experimental models that recapitulate human disease. And he is just an expert in this space. So thank you, Tom, for the time that you're spending with us this morning.
Of course, thank you so much for the invitation to present, and would like to first of all thank the sponsors, the donors, for their generosity in supporting this research program. This is really exciting for me personally because I'm sort of, as Mercedes described it, a little bit peripherally involved with tumor biology, but at the same time, some of the things that we've been studying for over a decade now have some relationship in and help to frame some of the questions related to tumor biology, as we heard about from talks from Michelle and others throughout the morning. And so I was initially asked to sort of present our work on some of the ex vivo models that we are utilizing, but I thought I couldn't really do it justice without giving a few words of introduction about where they really come from and what are the justifications and why do we really need these alternative models. So as you all know, the human brain contains billions of neurons distributed across dozens of functionally and anatomically distinct areas, including the areas that relate to speech, as you heard from Eddie. But ultimately, these neurons don't appear out of nowhere. They don't just, fall from the sky. They develop, as you heard from Mercedes, over many months of prenatal and postnatal development. And in humans in particular, this development is extremely protracted. And so fundamental and central to our understanding of where these cells come from is this very early neuroepithelium that surrounds the lateral ventricle of the developing human brain. And the cells that are located in that region are called neural stem cells or radioglial cells, and they are depicted schematically. This is what they look like, and they express these markers of stemness, including SOX2 and MKI67. Now, many of the mechanisms according to which the brains develop have been studied extensively in mice, and this is where most of our knowledge comes from. But even very early studies at the level of histology, comparative studies that compare mouse and human brain development begin to unravel some of the important differences in terms of the stem cell heterogeneity. So if you compare the mouse developing neocortex, the stem cells are primarily located to this ventricular zone, and this region is also present in the human, but in humans and also non-human primates and many other species, especially large brain mammals, tend to have this expanded outer subventricular zone region, which harbors these additional population of neural stem cells called outer radioglial cells that were described over 10 years ago. And these cells have a very distinct and unique morphology and dynamic cell behavior that involves this mitotic somal translocation, which is characteristic cell behavior for these cells. Using single cell transcriptomics and single cell approaches, we've been fortunate to be able to begin to characterize their unique molecular properties, including expression of transmembrane receptors that potentiate the growth factor receptor signaling that we published several years ago. And in the past couple of years, there's been a real emergence in these comparative studies that of brain tumor biology and especially of brain tumor stem cells that draw the sort of inspiration from comparative approach in trying to understand what's different between mouse and human brain development, and especially in relation to the stem cell heterogeneity that's related to that, and begin to unravel that these stem cell populations can also be found actually in both glioblastomas, and more recently, similar studies and observations have been related to medulloblastomas in that distinct regions of the rhombic lip of the human cerebellum develops quite differently than from the mouse rhombic lip, and the stem cell populations are differentially proposed to be differentially sources of stem cell populations in medulloblastoma. And so that really highlights this central concept that humans are not mice and that there are fundamental differences which drive us to our appreciation that in order to really develop better preclinical models, we really need to develop human-based models of some sorts. And so one of the areas which has become, very popularized in the recent decade, thanks to both the discovery of stem cells by Gail Martin at UCSF and also Shinya Yamanaka in his the seminal studies discovering factors that can reprogram patient fibroblasts into induced pluripotent stem cells, have really opened up a new field in our ability to model human brain development in a dish. These innovations have enabled us to culture human pluripotent stem cells routinely in the lab from many patients, and moreover, have added versatility in that these models can be used to actually to be derived from multiple different patients to model human genetic variability, specifically derived cells from patients with genetic mutations, such as in psychiatric disorders, and somatic, using genome engineering, we can introduce specific mutations in specific genes. So you can envision sort of introducing disease relevant or putative cancer driver genes into these pluripotent stem cells and begin to model their consequences on brain development. You can also derive these stem cells from multiple species, and there are a number of really wonderful studies doing that. And what we can do with these stem cells, of course, is we can actually subject them to some of the same developmental instructions as normal developing stem cells experience on their way to becoming, for example, the cerebral cortex or the medial ganglionic eminence, which you heard about from Mercedes. And these two stem cell populations will give rise to different neuronal subtypes. But we can also actually pull these cells together and grow them in three-dimensional spheres, and they will actually derive, they have this immense property to self-organize around these beautiful neural rosettes. This is what a cross-section through an organoid looks like. And you can actually see these cells stained for white, this is the staining for SOX2 actually. And you can see that these neural stem cells begin to self-organize into these higher order structures called neural rosettes that actually resemble many of the early stages of brain development in vivo. So if you compare these stem cells lining a adhesion belt, this is exactly what neural stem cells do in normal human brain development. But in addition, you can also begin to appreciate that these human stem cell derived organoids begin to elaborate these stem cell niches into these cells that are ventricular. And in fact, when you compare the dynamic cell behavior in these models, the behavior that describes and distinguishes these outer radioglial cells is actually recapitulated also in brain organoids. And so these models are emerging as a potentially very useful way of modeling human neural development and gaining access to human genetic background and human genetic mutations. These models, of course, have their own limitations, and in particular, one that's been sort of highlighted in recent studies has been this notion of emergence of integrated cell stress as a consequence of cell culture. So because these cells grow, they don't have access to vasculature, they don't have access to the immune system. There's only so many times you can feed them and exchange the media. As a consequence of all that, these cells upregulate many markers of hypoxia. Thankfully, these hypoxic conditions and this integrated cell stress is not an irreversible process. You can take these organoid cells, transplant them into a mouse, as SCID mice, and you can actually rescue the hypoxic condition. So you can see that the markers of hypoxic stress are downregulated post-transplantation with quite a short period of time. Now, the caveat of this particular approach is that it's not particularly scalable because you cannot really do this at scale. You can inject hundreds of mice, but that's not particularly useful. So we really need to find a way to overcome this issue and develop models that can be routinely used in the lab. So one approach that we've been taking and we've been very happy with is actually taking these brain organoids that they grow in a dish and they eventually would normally develop hypoxia, except that we slice them pretty much as organotypic slices, and then we culture them at the air-liquid interface. And that allows these cells to actually develop more sort of normal metabolic properties and to elaborate their organization in vitro. And a recent study from Guoli Ming's lab has actually also demonstrated that this procedure allows the cells to actually have a better access to to oxygen, and that allows them to reduce some of the cell stress. So this is really promising, and there's a lot of excitement about applying these models to study the consequences of specific genetic mutations, including in the context of brain tumors, as you heard about from Mercedes in the context of tuberous sclerosis. But there are still limitations that I sort of hinted at earlier, which is that, okay, even if you overcome the metabolic cell stress, there is still a lot of cell types that are missing from brain organoids, including in particular when you compare in vivo developing human brain to these ex vivo neural lineage derived organoids, you're missing some of the important components, including microglial cells, endothelial cells, and other cell types. And these cells, of course, are critically important to normal human brain function. Blood vessels are important, not just to deliver the blood and everything that comes with it, but they actually also secrete factors that promote and interact with neurons. And you've heard a little bit about the immune system. That also plays an important role in disease. And in particular, when you look at brain tumors, the brain tumors very frequently, as you heard about from during Michelle's talk, have this immense capacity to co-opt the environment for their own purpose. Okay, and so that means, as you heard earlier, both co-opting the neural activity, co-opting the oligodendrocyte myelination, but also actually promoting their own vascularization. And I think that's really remarkable. Moreover, they can interact with the immune cells, and particularly the macrophages in brain tumors, to actually push them towards a state that will only promote their growth. So they're really, these cells that would normally play a protective role can be sort of co-opted into bad players that will actually promote tumor growth. But these cells inherently don't develop in organoids. And so what Galina Popova, who is here with us in the audience, did in my lab, has taken actually on a very challenging project to ask the question, can we reconstitute this immune environment into brain organoids? And so we derived these brain organoids from pluripotent stem cells, and then we took primary microglial cells derived from primary brain tissue, and we just simply transplanted them, added them to brain organoids. And indeed, within a few days, these cells integrate very nicely into the organoids. They send their processes, and they really colonize the entire brain organoids throughout, and they can be maintained in vitro for quite a long period of time. So then Galina wanted to know, well, did it matter in any way to neural development? What do these microglia, for example, do in brain organoids? And what she found was that these microglial cells very closely interact with neural stem cells. They can actually reduce some of the effects of cell stress, including DNA damage response. And I think that will be quite interesting in thinking about how mutations can accumulate over time of culture, and particularly as it may relate to the occurrence of mutational burden when we start thinking about modeling brain tumors in a dish. She also placed these brain organoids on multi-electrode arrays, which is a technology that Mercedes also introduced in her talk, where an organoid is placed directly on these electrodes in a dish. And what she found was that while looking at the spontaneous activity in brain organoids, you can see sort of occasional action potentials firing. These are these red lines. When she adds microglial cells, these organoids actually begin to develop these more synchronized patterns of electrical activity, suggesting that reconstitution of more physiologically relevant milieu of cell types in developing brain organoids is really critical to our ability to model these more complex network properties. Another cell type that I sort of hinted on earlier that I think is really quite important is also the vasculature. So vasculature, on one hand, plays this role in delivering the blood and all the oxygen and nutrients that come with it, but also it delivers cell types that themselves can actually promote brain development in many ways. And these cells, again, because they don't come from the neural lineage, we have to derive them separately and then somehow reconstitute them into brain organoids. And so thankfully, people have actually developed these culture conditions that can turn pluripotent stem cells directly into vascular endothelial cells, pericytes, and other cell types of vasculature. And Jaden Ross in the lab, who was a trainee earlier, together with Ethan Winkler, have developed or have tried to reconstitute these vascular cells into brain organoids. But they first tried to just simply, same as with microglia, just transplant them and see what happens. And in fact, that was a complete failure. And probably because this is not how the vasculature normally develops. It sort of develops from the vascular plexus and then it grows in. Jaden also tried just like physically injecting them into brain organoids, and that was a complete failure as well. And then Jaden came up with this idea, well, what if I actually took these cells and tried to mimic what happens normally in vivo, embed them into a matrigel, into a hydrogel, and then let them sprout and then put them next to the organoid. And that's exactly what allowed Jaden to actually develop these organoids that actually contain this beautiful vasculature highlighted here in the red in these organoids. And there are many aspects of normal development that are actually recapitulated, including you can see the emergence of these pericytes that are actually shown here in cyan that line up these blood vessels, these endothelial cells marked in red. These vascular endothelial cells develop tight junctions marked here by Claudin 5. And they even begin to form these angiogenic sprouts, which are really quite characteristic of the developing vasculature. And when then Jaden started to look at what happens at the neural development, in fact, incorporation of these vascular endothelial cells into brain organoids promoted the emergence and expansion even further of these neural stem cell populations that we found to be present. And so we really think and are excited to think about opportunities in which we can utilize some of these technologies to begin to compare the interactions that occurred during normal development between microglial, endothelial, and neural stem cells and how they may become co-opted in the context of brain tumors. And then there is another opportunity, more broadly thinking about organoids don't just need to be derived from pluripotent stem cells, they can be derived also directly from patients. And this is another project which we Galina started collaborating on with Sean Hervey-Jamper here, and in being able to gain access directly to patients' tumor tissue and culture and begin to culture these tumor tissues exactly like brain organoids. And that's really been a very successful endeavor. And Galina has been consistently deriving these brain organoids in vitro. She has also tried several different ways of innovating on culture methods, including asking the question whether culturing them in different metabolic conditions can regulate the stem cell growth. And in fact, when she cultured, she co-opted some of the same lessons of culturing organoids on air-liquid interface that I told you about earlier in the talk versus 3D sort of metabolically stressed organoids. And in fact, culturing them at the air-liquid interface really massively expanded their proliferation. So we're very excited about thinking about how we could utilize these experimental models to begin to test some of the hypotheses that might emerge from measuring some of the metabolic factors directly in patients and then subjecting these neural stem cells directly from patient brain tumors to these conditions in ex vivo models. Finally, I just wanted to mention another approach which Galina is taking in developing this model further. She's actually fusing these brain tumor samples with brain organoids to begin to look at the dynamics and also mechanisms of tumor metastasis, and shown just over here as a fusion of a patient brain tissue. And you can begin to see, it's sort of hard to see, but in a section, you can begin to see these cells, these glioblastoma cells, labeled with a dye this is the original tumor that was fused to an organoid, and you can begin to see some of the cells sort of metastatically entering into brain tissue. And so in summary, we're very excited about the many opportunities that these approaches create. And we really think that this is quite important in anchoring some of our studies, especially when it comes to stem cell proliferation mechanisms in basic fundamental understanding of human brain development and cellular heterogeneity, and thinking about how to leverage some of these stem cell derived organoids to model specific genetic mutations, develop new experimental models, but then also to begin to utilize access to primary brain tumor tissue to begin to study, to develop new preclinical models for testing therapeutics. And this is really an emerging field that's becoming really quite exciting, and especially with the refunding of the California Institute for Regeneration Medicine, it will be hopefully more commonly used as an experimental model for modeling tumor growth and clinical applications. And with that, I would just like to thank all of you for the invitation, all our collaborators. And if you have further questions, please refer them to Galina, who's right here with us in the audience. So happy to take some questions.
Great talk. You know, as we're transitioning over, I have to ask about low-grade organoids. Any thoughts there?
Yeah, so that's a fantastic question. We've only really begun to look at and culture these models, but I think comparing brain organoids as they grow from different types of mutations from different types of patients, I think will be a really useful approach to begin to compare the dynamics of cell growth and metastasis. So I think there is just so much to do in this space, and we're just really excited that the technologies to culture these cells in a lab are becoming more commonplace. So